So today, our seminar speaker is Dr. Adriana Romero-Olivares. Adriana is an assistant professor in New Mexico State University. She did her undergrad in Autonomous University in Baja California, and then did her master's and PhD at University of California, Irvine. Adriana's lab focuses on how microbes adapt and evolve in response to global climate change, and she works with several soil fungal species in her lab and is interested in how drought impacts fungal growth and development. Okay, Adriana, you can probably start your talk.
Yep, thank you so much for inviting me. So today I'm going to talk about fungal responses to climate change and consequences to the carbon cycle. And I'm going to give a general introduction to my talk, just to provide some background and context to it. Okay, so when I decided to study biology, I did so because I was fascinated by landscapes like this one, with a lot of visible biodiversity, a lot of plants, a lot of animals. And I was born and raised in the desert, so these types of ecosystems were very foreign to me. But back then, I never really thought about what was beneath all this visible biodiversity. But as I moved forward in my degree, I started to learn about the importance of soil and its very, very large invisible biodiversity, and how essential this biodiversity is to keep ecosystems working. So, for example, in this image, this is a relatively small piece of land, and even though it's relatively small, there's approximately 50 kilometers of mycelium of approximately 500 different fungal species. There's approximately 100 billion of bacterial cells of approximately 10,000 different species. There's hundreds of species of algae, thousands of different protozoa, and you know where I'm getting. So there's a lot of invisible biodiversity that inhabits soils. And in my work, I focus mostly on fungi, more specifically in soil fungi. So soil fungi are fungi that live all of their life or a part of their life in the soil. And there's a lot of soil fungi. In a single gram of soil, there can be thousands of different species of fungi. And if you're lucky, sometimes you can see them. So for example, in this picture, you can see a piece of wood that has been completely colonized by mycelia. So all of these white threads that you see here, that is fungal mycelium. In this other picture, you can see the type of fungi that we typically think about when someone says the word mushroom. It has its head and it has its stalk. And in this other picture, there's a quote unquote a less traditional type of mushroom. The correct term would be a fruiting body, but it's still a fungus, and it's still visible to the naked eye. But the great majority of fungi are microscopic their entire life cycle. They never make these fruiting bodies. They don't create those very thick mycelial threads. They're invisible, and there's no way that we can see them with our naked eyes. Sometimes we can see them if we isolate them using microbiological techniques. So, for example, all of these petri dishes that you see in the picture, these are fungal isolates that we isolated from desert soils. And then there's some other fungi that we have never seen. We know they exist because their DNA sequences come up in our studies, but we don't really know what species it belongs to. We don't really know how that fungus looks. And to jump into my research topic a little bit more, as you can imagine, so fungi, just like any other organism on Earth, they're extremely vulnerable to changes in the environment. But because they're invisible to the naked eye, it's really difficult to study them. So for example, when you see this picture, there's things that you can say about it. On the right side of the picture, you can say that that ecosystem is probably stressed. The ground looks eroded, it looks unhealthy. The plant doesn't have any leaves. So you can say that side of the picture is probably a stressed and unhealthy ecosystem. And on the other side of the picture, you can say the opposite. The ground looks pretty healthy, has a lot of green grass. The tree has leaves, and it looks just in general pretty healthy. But what could you say about the microbes in these ecosystems? And you can't really say anything. You can assume certain things based on the condition of the ecosystem, but you can't say for sure how the microbial community is doing. And there's just certain things that for us microbiologists, we simply cannot do with our study system. So for example, we cannot follow fungal life cycle because it's impossible to count the progeny of fungi. We don't even know the growth rate of fungi in the soil. There's no historical record on the distribution of fungi except for very few mushrooms, and these records are very specific to certain parts of Europe. And there's other reasons why studying microscopic fungi is really difficult in ecology and evolution. But despite this difficulties, it's really important that we study them. And it's really important that we study them because fungi are mediators of biogeochemical cycles. It means that they're responsible for keeping nutrients and elements flowing in our ecosystem, specifically between land and atmosphere. So for example, in this picture, you can see how fungi are mediators of the carbon cycle. So a very simplified carbon cycle, starting with sunlight and carbon dioxide, plants photosynthesize and produce biomass in the form of leaves and in the form of wood. And then this biomass eventually die and falls onto the ground. And in the ground, they are decomposed by decomposer fungi and by other microbes and other organisms in general. A portion of the carbon that was once leaves is organically bound in the soil. A portion of the carbon the fungi use for themselves to build their biomass, and a portion of the carbon is released back into the atmosphere as CO2, where it incorporates back into the atmosphere and where trees can eventually use it to photosynthesize, completing a round of the carbon cycle. And I do want to pause here and mention that there's another group of fungi. These are mycorrhizal fungi. These make symbiotic relationship with plants, but in my work, I focus mostly on free living fungi. So all of the work that I'm going to be showing is only about free living fungi. And all of this is really important because soils are not only the home of soil fungi and of other organisms, but they're also a carbon storehouse. There is around twice as much carbon stored in the soil compared to what's already available in the atmosphere. And because soil is a storehouse of carbon, it can also be a source of carbon dioxide. And it's for the best that the carbon that's stored in the soil stays there, because if it's released into the atmosphere, concentrations of CO2 would increase. So in a few words, if a lot of the carbon that's stored in the soil is released into the atmosphere as carbon dioxide, the greenhouse gas effect and global climate change can worsen. And all of this is really important because the molecular machinery that microbes use to decompose and to store and to return CO2 into the atmosphere is extremely sensitive to environmental change. So environmental change and impacts to soil fungi can result in all sorts of different ecosystem scale consequences. So for example, it can result in millions of dollars lost in crops if environmental change happens to benefit the increase in abundance of pathogenic fungi that infect crops. As I just mentioned, it can result in increases in greenhouse gas emissions, and it can even result in changes in the microbial community that can result in the emergence of pathogenic fungi that infect humans. So with all of this in mind, I went to the Boreal forest in Alaska, and I was especially interested in studying this ecosystem, not only because I was fascinated by these ecosystems since always, but also because Boreal forests store approximately 510 billion tons of carbon in the soil, and because changes in temperature brought by global climate change have been really drastic at these latitudes. So in this map, this is a map of the state of Alaska. You can see the location where I worked in is Delta Junction right here. And according to this map, all of the state of Alaska, including the area of Delta Junction, has been significantly affected by changes in temperature. So specifically, the area of Delta Junction has experienced an increase in 1.7 to 2.2 degrees Celsius in the average annual temperature compared to the past 50 years. I conducted research in this in a field experiment. At the time that I sampled, the experiment had been ongoing for over 10 years, and it consisted of 5 greenhouses in the middle of the forest, just like the one in the picture. And in this other picture, you can see each one of these thumbtacks represents where each one of the greenhouses were. And these greenhouses, these are a pretty remote site. So the only way that you could emulate warming is using warming the soils passively using greenhouses. Very far away from electricity, so it's not possible to have heating cables or any other active warming device. So we had these greenhouses that passively warm the soil and the air inside them. They increase the inside air temperature by 1.5 degrees Celsius, so around the increase in temperature that the area of Delta Junction is actually experiencing. And it also increases the temperature of the top 10 centimeters of soil by 0.5 degrees Celsius. And because of that, evapotranspiration is higher inside the greenhouses. And this causes that the inside of the greenhouses is 22% drier compared to the outside of the greenhouses. So our treatment in this field experiment were the inside of the greenhouses, and then control plots were at adjacent plots, just somewhere around the greenhouses. And maybe you're thinking that 1.5 degrees Celsius or 0.5 degrees Celsius is not really that much. How can that possibly make any changes or cause any changes in the soil? But here you can see how different the control looks like compared to the inside of the greenhouses. So this is the outside of the greenhouses right here. A lot of moss, a lot of plants, a lot of mushrooms, and then this is the inside of the greenhouses. So pretty dry, hardly any mosses, hardly any plants, one mushroom. So in this field site, I wanted to know what's happening with the soil fungal community under warming and drying. Where are they allocating resources, and how does this reflect in the cycling of carbon? So to answer these questions, I carried out 2 different experiments. The first one was soil metatranscriptomes, where basically I extracted RNA from the soil, and from control plots and from treatment plots, and then analyze metabolic maintenance genes, also known as COG genes, and decomposition genes, also known as Casii genes. And then the other experiment was measuring decomposition rates using litter bags. Probably most of you are familiar with this method, but if you're not, it's really simple. You just use litter bags like this one that you can see here. You collect senescent spruce needles. So in this case, we use spruce needles because these were the trees that are most common in the field site. So you collect senescent spruce needles, a specific amount. In this case, we use 2 grams, and then put those 2 grams inside the litter bags, deploy them on the forest floor, and then let decomposition happen. And so the way that you measure decomposition is by measuring mass loss, which we did for 1.8 years. So what did we find? So we found that warming and drying favors melanized, pathogenic fungi, and desiccation-tolerant fungi, such as crust and lichens. So by favor, I mean that these groups of fungi were the ones that were more active under warming, and by more active, I mean the ones that were transcribing the most. So again, a lot of melanized fungi like Cellaria species, like Janula, a lot of pathogenic fungi, mostly plant pathogens like fungi in the order of the Diaportalis, some penicillin species, and then desiccation-tolerant fungi like a lot of crust fungi and a lot of lichenized fungi. Metatranscriptomes also revealed a trade-off in resource allocation. So in this graphs, in the x-axis, you'll see metabolic genes here and decay genes here. And then in the y-axis, you'll see total gene transcripts here, metabolic gene transcripts, and here decay gene transcripts. And then control plots are in gray and warm plots are in black. And as you can see, the trends are the exact opposite, meaning that under warming, microbes allocate more resources to cell metabolic maintenances and less resources to decay, whereas in control conditions, you see the opposite, more transcripts allocated to decay and less transcripts allocated to cell metabolic maintenance. And I do want to point out that, transcripts are just that, they're transcripts. We can't be sure that all of these transcripts, which are messenger RNAs, are going to mature and are going to be translated into enzymes. And we also can't be sure that these changes will have an impact at the ecosystem level. However, our field decomposition experiments supported our metatranscriptomes finding. So in this graph, you'll see the different time points where we collected the litter bags in the x-axis, so decomposition time. And then in the y-axis, you'll see mass remaining as percentage. And then control plots are white bars and warmed plots are black bars. And you can see that for all collection times, the percentage mass remaining was always higher in the warm plots. So this means that decomposition in warm plots was lower compared to decompositions in the control plots. So indeed, what we found in our metatranscriptomics data, that underwarming microbes are investing less in decomposition, was supported by our field observations because there was less decomposition underwarming compared to control. So to answer the questions, what's happening with the soil fungal community underwarming and drying? Pathogenic, melanized, and desiccation-tolerant fungi were more active underwarming. Where are they allocating resources? They are allocating more resources to cell metabolic maintenance and less resources to decay. And how does this reflect in the cycling of carbon? There's less decomposition underwarming. So a question that I get very often is, are these results good or bad? So I think that the answer is that it's a little bit of both. So first, it looks like it's good because there's less decomposition, and that probably means that there's less CO2 that's being returned into the atmosphere. But shifts in the fungal community are concerning, especially the increase in pathogenic fungi. And we have to remember that other microorganisms and plants and other organisms in general acquire their nutrients from fungal decomposition of organic matter. So less decomposition results in less nutrients available for plants and animals. I do want to point out that these results are not unique to our field experiment. There's other warming experiments that have found very similar results. I'm not going to have time to talk about that, but I summarized that in a meta-analysis published in Soil Biology and Biochemistry in 2017. And I also want to point out that the decomposition results that I presented are really nuanced, and I'm also not going to have time to talk about this today, but you can look at those results in the 2017 publication in SWaN, where we look at which fractions of carbon are decomposed the most under warming and drying and which ones are not. So in general, decomposition is lower under warming, but there's certain fractions of carbon that their decay is accelerated with warming. So the conclusion is that it's too soon to tell, but what we know is that global climate change is likely to affect the fungal community with consequences at the ecosystem scale, such as causing disruptions to the carbon cycle. And I forgot to mention that this was part of my PhD work at Kathleen Treesteter's lab at the University of California, Irvine. And for my postdoc, I continued to work in northern ecosystems. I worked with Dr. Sarita Fry at the University of New Hampshire. And this time I moved a little bit south. So I started to work in temperate forests of North America, specifically in the state of Massachusetts. So in this area, soil carbon stocks are also very large. and the mean annual temperature increase that's predicted for this area is of 0.3 to 6.4 degrees Celsius by the year 2100. And this area has also suffered historically of elevated deposition of reactive nitrogen. And although some environmental policies have decreased the deposition of nitrogen in the past couple of decades, this area is still receiving elevated rates of nitrogen deposition, and it has been historically a good model of the effects of nitrogen deposition for temperate forests. So this time, I was working at Harbor Forest LTR, and here there's a global change field experiment that is using soil warming and nitrogen deposition as global change drivers. And at the time that I sampled here, this experiment had been ongoing for 10 years. And it consists of plots that are actively warm. So we're closer to electricity now. So there's heating cables below ground that actively warm the plots. There's other plots that are fertilized with ammonium nitrate, and then plots that are both warmed and fertilized. And then plots that are left as is, which serve as controls. And you can see here what the setup looks like. So control plots, heated plots, nitrogen plots, and then both heated and nitrogen plots. And in this field study, I wanted to explore the topic of soil volatiles and how these may change in response to global change drivers. So not fungal specific, just microbial specific in general. So for those of you not familiar with volatile organic compounds, which I will refer to as VOCs, these are organic chemicals that vaporize at room temperature. And there's many different types of VOCs, and some of them are ecologically important because they mediate relationships between microbes and plants, plants and plants, plants and insects, and so on. And some other VOCs are important for atmospheric budgets because, for example, they can be precursors of ozone. And microbial VOCs specifically have not been deeply explored in microbial ecosystem ecology. There's a lot of work done with plant VOCs and VOCs budget at the atmospheric level, but not a lot of microbial VOCs and how these relate to ecosystem ecology. And because of that, they have been labeled as a grand challenge of terrestrial ecosystem ecology because we know so little about them. And a perfect example of what a soil VOC is, if you've ever thought about, oh, it's gonna rain and it already smells like rain, that smell of rain is geosmin, which is produced by many soil microorganisms when there's like some humidity. And like geosmin, there's many other VOCs. Some of them smell and some others don't smell. So here I wanted to address two questions. Do soil VOC profiles change under warming and nitrogen fertilization? And what is the role of VOCs in microbial communities under global climate change? So to answer these questions, I collected VOCs from the soil warming and nitrogen field experiment at Harbor Forest, and I sampled in 2018 and 2019. And I used this setup that you can see here. I modified these light core and modified it in a way that I was able to insert these tubes that are called 10x tubes where the VOCs are absorbed. And then after the collection, using GC mass spec and some specialized software, we were able to determine which VOCs were coming from the control plots and which VOCs were coming from the treatment plots. Okay, so what did we find? Here you're going to see two graphs. This one is from 2018. This one is from 2019. In the x-axis, you'll see the control, and then the warming, the nitrogen, and their interaction. And then in the y-axis, you'll see total VOC emission. So we found that emission rates were very different by year. In 2018, there were significantly higher VOC emissions in the treatments compared to control plots. But then in 2019, we saw that there were lower emissions and that there were no significant differences on the emissions between treatments and control plots. So then to visualize these results, I conducted an NMDS. And as you can see, the different colors are the treatment. And then the different figures are the different months that I sampled in. So August are circles, June is triangles, October squares, and then September crosses. And as you can see, there is no clustering based on treatment, and there's also no apparent clustering based on month. However, there is a couple of two major clusters which correspond to the two different years that I sampled, which makes sense considering that we did see a very strong annual difference in the emission rates of VOCs. Something really interesting is that we found some very specific signature VOCs. So these are VOCs that are or were consistently found in specific treatments, regardless of the year, regardless of the month. These VOCs were always found consistently in the treatments, and although at low emission rates, we found them throughout all samplings in all in both years. So here you'll see for different graphs, treatment is in the x-axis, and then in the y-axis, you'll see the emission rate of the different VOCs. So alpha-pinene, beta-thujone, beta-caryophyllene, and 2,4-dimethylheptane. And what we saw is that, for example, alpha-pinene had significantly higher emission rates in warming plus nitrogen compared to all other treatments. Beta-thujone had significantly higher rates on nitrogen and warming plus nitrogen compared to control. Beta-caryophyllene had significantly higher emission rates in the interaction of warming and nitrogen compared to control. And then 2,4-dimethylheptane had significantly higher emission rates under warming and warming plus nitrogen compared to control. And even though we don't know who's producing these VOCs, we know they're coming from the soil. And they have been widely reported as being produced by bacteria and by fungi. And all of them have been reported to have strong antimicrobial properties. So in my work, I've done a lot of culture work, and most of my research is always complemented with culture work. And one of the things that I'm really interested in looking at is fungal traits and how these traits change in response to different environmental stressors. And during my last few months of my postdoc, I was confronting fungi and bacteria isolated from these field sites. And I was using different temperatures and different concentrations of ammonium nitrate. And I wanted to get a sense of how the interaction via VOCs changed under different environmental conditions. So I use these type of petri dishes. So these type of petri dishes have a division in the middle, and you inoculate, in this case, a bacteria on this side and then a fungus on this other side. And then this is what I mean by confrontation. And although it looks like the division is completely straight, if you look at the petri dish at an angle, you can see that there's a change in the height of the division. So then when you put the cover of the petri dish, this side of the petri dish is completely covered, but then this side allows for gas exchange, and that way you can confront the fungi and or the microbes, and then you can see if there's any sort of inhibition, and the inhibition that may be going on, you assume is via VOCs because there's no touching between the microbes. So like secondary metabolites like antibiotics cannot be passed because of this division, but there is gas exchange. So one of the things that I saw consistently is that the bacteria that was isolated from the warming and nitrogen plot usually had this inhibition effect on the fungi, to the point where the bacteria would first inhibit the growth of fungi and then would sort of like jump to the other side of the petri dish to start colonizing the fungal section. I also confronted fungi with fungi, and this effect was not really that strong between a species of fungi against another one. In some cases, there was like jumping of the mycelia, as you can see here, but I didn't really see that strong inhibition effect that I saw between bacteria against fungi. This in itself is really interesting because in this field site, bacterial biomass has increased in abundance in response to warming and nitrogen fertilization, and fungal biomass has decreased. Unfortunately, this work was cut short by the pandemic and I didn't get a chance to finish up. But I do have an undergrad working on finishing up these confrontations, so hopefully we'll have some interesting results in the next few months. So to answer the questions, do soil VOC profiles change under warming and nitrogen fertilization? Yes, but in our data, there was a very, very strong seasonal effect. And what is the role of VOCs in microbial communities under global climate change? And that is to be determined. So I think that VOCs may be serving as microbial biomass, may be serving as control of microbial biomass under global change drivers, but this is still something, a topic that I'm still exploring, and I am actively looking for collaborators for this study because I require someone with the level or with the level of expertise that you need for the collection and analysis of VOCs. So if you're interested in collaborating, please reach out. Okay, so in the last few minutes of my talk, I want to talk about one of the ongoing projects of my lab. So, I talked about boreal ecosystems and temperate ecosystems, but what about dryland ecosystems? So these ecosystems are super important. They cover approximately 40% of the Earth's surface. They are home to more than 2 billion people. So that means that 1 in 3 people in the world inhabit a dryland ecosystem. They are characterized by being water limited, sparse vegetation, although not obvious from these pictures in my opinion. So this is the Chihuahuan Desert in New Mexico. This is right outside New Mexico State University where I work. And then this is the Sonoran Desert in Sonora, Mexico, where I was born and raised. And another thing that's very typical of these ecosystems is that they experience large temperature fluctuations in a day. So it can be really hot at noon, and then it can be really cold at midnight. And these ecosystems are also super important for food and nutrition security of the whole planet, because up to 44% of the world's cultivated systems are located in drylands. In addition, a lot of drylands serve as rangeland for livestock. And finally, drylands are super diverse and house, many animals, many plants, and many, many microbes. And like any other ecosystem on Earth, drylands are affected by climate change, particularly by changing rainfall patterns and land degradation. So here you can see that this is the U.S., and you can see that the southwest of the U.S., so where most drylands are in the U.S., they are experiencing the largest percent change in precipitation. So you can see that all of this area that is beige and yellowish, it's going through this percent change in precipitation. So it's experiencing 10, 20, and 30% less precipitation compared to the last couple of decades. And in this figure right here, so this is the Chihuahuan Desert right outside of Las Cruces, where I live. So this is what the Chihuahuan Desert looked like in 1920. So you can see there's a lot of grasses. These are native grasses and very few shrubs. But in the past few decades, there's been this transition of the ecosystem. So the ecosystem is transitioning towards a woody, plant-dominated landscape. So you can see very few grasses and many, many shrubs. And although this picture is from 20 years ago, this transitioning of the ecosystem is pretty much still happening. And these changes in ecosystems are often interpreted as desertification. So basically the broad scale conversion of perennial grassland to dominance by xerophytic woody plants and the associated loss of soils and biological resources, including biodiversity, is known as desertification. And in this ecosystem specifically, so large number of domestic livestock in combination with extreme multi-year droughts have been devastating for native grasses and have allowed native drought-adapted long-living shrubs to dominate throughout the region. And the Jornada Basin is located in southern New Mexico, so right here. Is in the northern extent of the Chihuahuan Desert. So the Chihuahuan Desert is huge, as you can see, is the largest of the North American warm deserts. And we're working here at the Jornada LTR. And as I mentioned, this region has undergone large shifts in the relative dominance of grasslands and shrublands over the past century. And the dominant vegetation at the moment, depending on where you are in this, in the Jornada LTR, is a huge LTR. So depending on where you are, you can find different patches of vegetation. So there's still some patches where livestock has been excluded, where you can still see a lot of native grasses like here and like here. But the rest of the Jornada looks mostly like this. So it's very shrubby. Because there's a lot of livestock activity right here. So the shrubs are usually native woody plants such as mesquite, tarbush, and creosote bush. Okay, so here in the Jornada, there is a global change experiment where this experimental site is a fully factorial block design with 9 blocks of 3 treatments. So drought, disturbance, and then drought plus disturbance, and then control plots. Drought is completed with rainout shelters that you can see here, which impose 80% reduction in incoming precipitation. Disturbance is created using vehicles with rototillers to impose surface disturbance that mimics vehicle damage, but also extensive grazing. And this field experiment is fairly new. It was established a couple of years ago. So here, we wanted to know, how does the fungal community respond to global change drivers, drought, disturbance, and its interaction? And to answer this question, we sampled soils from control and treatment plots 1 year after the onset of the experiment. And then within each treatment, we sampled in 2 different locations, under vegetation and then in the bare soil between shrubs. We wanted to do this because the loss of native grasses and increase of woody shrubs has created a patchy landscape that is expected to become more common in the next few years. And we expected the community under vegetation to be very different from the community at the interspace. And we wanted to make sure that we captured responses of both. So we extracted DNA, conducted metabarcoding with fungal-specific primers, and then sequenced the ITS2 region. And the main finding is that the soil fungal community clustered by location, and we didn't really find any treatment effect. So here you can see the treatments in different colors, and then the location, interspace in circles and under in triangles. And you can see that there's 2 major clusters with a little bit of an overlap, and the clusters correspond to location. And you really don't see any clustering based on treatment. We found that just in general, dryland soils were dominated by Basidiomycota fungi. This is kind of different from what most other studies have shown, although high-throughput sequencing has recently shown that Basidiomycota are pretty common in dryland ecosystem. We just didn't have the proper tools to look into this before. However, Basidiomycota fungi are mostly yeast. So there's a lot of Basidiomycota fungi that have a yeast form like this one, Rhodosporidiobulus, Phylobasidium, and Naganitia. Oh, I forgot to mention. So you'll see 2 graphs, interspace and under vegetation. In the x-axis, you'll see the treatments, and in the y-axis, the relative abundance in percentage. And you can see that Basidiomycota are the taxa that dominates the soils in general. However, Chytridiomycota here, this right here, this orange right here, are more common in interspace soils compared to under vegetation. And then you can see that unknown fungi are also slightly more common in interspace soils compared to under vegetation. So we did find some differences, mostly based on location, but not really between control and treatment plots. The fact that we found chytrids, these are some primitive fungi that disperse via zoospores in the interspace where it's super dry, was really an interesting finding, but just looking at the literature, it seems that other people have found similar results. These graphs are the same, so treatment in the x-axis, relative abundance in the y-axis. So we found that Saprotrophs were the most common functional group in dryland soils. You can see here that Saprotrophs, this intermediate blue, is the most common. Pathotrophs are light blue, and then dark blue are symbiotrophs. And you can see that Saprotrophs are the most abundant in both interspace and under vegetation. When we break down these different trophic modes into more specific functional groups, we see that still general Saprotrophs are the most common type of fungi in these soils. We can see that plant pathogens are somewhat more common in interspace soils compared to under vegetation, and that animal pathogens may be slightly more abundant under vegetation compared to interspace. And once again, we found differences between location, but we didn't really find differences between control and treatment soils. Finally, we looked at the fungi and how, based on their morphology, and we found that microfungi were more common in interspace. So microfungi are fungi that have microscopic spore-producing structures like molds, mildews, rusts. So Alternaria, this right here, is a microfungi. Staphylococcus is another microfungi. And we saw that those were very common in interspace soils. And we saw that yeast, again, Basidiomycota yeast like the Naganitia and Phylobasidium were very common mostly in under vegetation, but also somewhat common in control, drought, and the interaction of drought and disturbance. And even though some of these changes look very striking, they were not significantly different because there was a lot of variation within each one of the plots. We found, because of that global change drivers did not affect the richness or diversity of fungi. So here you'll see, again, interspace and under vegetation plots, treatments in the x-axis, and then species richness in the y-axis. And you can see that we didn't really see any significant differences except between control and disturbance under vegetation. But you can see that species richness, the variability of species richness was super high in the different samples, which I think contributed to the lack of significant differences. The Shannon Diversity Index, which measures the diversity of species in a community, also showed no differences in diversity between treatments compared to control. We carried out indicator species analyses. So these indicator species analyses shows you which species are preferentially occur under specific conditions, and most of them are found in high abundance compared to others. And we found that species with black arrows were very common in interspace, and red arrows were very common in under vegetation. And the takeaway of this slide is that indicator species in treatments are mostly saprotrophic fungi that can become pathogenic. At the moment, we're analyzing metatranscriptomes to investigate if there are differences at the transcription level. So maybe there's no difference at the community level, but at the transcription level, we may find something. We're isolating fungi from control and treatment plots. We've successfully isolated multiple strains of Naganitia, Phylobasidium, and Ceratobasidium. And we are actively trying to isolate more species of Wallemia and Geastrum. All of these were species that were very common in our data set, and we're interested in looking at their physiology a little bit more. Wallemia is an extremophile organism, which was very common in our soil sample, so we're interested in looking at them. And then Naganitia, Phylobasidium, these are saprotrophic yeasts that can become pathogenic to humans. So how does the fungal community respond to global change drivers, drought disturbance, and its interaction? So it seems the fungal community in the Chihuahuan Desert seems to be resistant to global change drivers after 1 year of exposure to drought and disturbance, but it may also be too soon to tell. We are complementing our study with other analyses to see if we find anything significantly different that may show that the community is not that tolerant to global change drivers. So with that, the summary of my talk is that global change is affecting fungi from genes to ecosystems with repercussions to biogeochemical cycles. I hope that I've convinced you that fungal responses to environmental change impact biogeochemical cycles. For example, it can cause disruptions to the carbon cycle, and that studying fungal responses to global climate change can allow us to better understand mechanisms behind ecosystem scale observations of biogeochemical cycles. And with that, I want to thank you for your attention. I want to thank the University of California, the University of New Hampshire, New Mexico State University, all of my funding sources from both Mexico and the US, my mentors, Dr. Sarita Fry, Dr. Kathleen Truceder, and all of my collaborators and students that have helped out throughout my PhD, my postdoc, and now as an assistant professor. And with that, I'd be happy to take any questions if there's any.
I have a question about the VOC work that you've done. In the Petri dish, the experiment where you saw like the inhibition of the fungal growth, did you look at what is the mechanism of that inhibition?
No, I didn't look into that. So that was sort of like the initial set of experiments where I just wanted to see if there was any sort of inhibition via VOCs. The next step of that project was to actually look at the VOCs that were emitted, which I didn't get to do. But no, I have no idea what the mechanism may be.
Hi, Adriana. Thank you so much. That was a great talk. I have one related question to what Arunima was asking about the divided Petri dish. Very intriguing experiment. So anyway, so did you also try the direct co-culture of those two fungal and bacterial species?
No, actually, that's a really great point. No, I did not try any co-culturing, but thanks for bringing that up. I have a student that is sort of like finishing up with this, and it hadn't occurred to me to do that. So that is something that
you know,
we should
kind of like not control per se,
Yeah.
right? But kind of to see the either the worst or the best case scenario, right? So,
Yes, yeah, exactly.
Yeah, great.
Thank you.
Okay, thanks.